Hi everyone, it's me again. My name is Dr. Chris Murphy. I'm a laser plasma physicist and I'm hosting this University of York School of Physics, Engineering and Technology webinar series. So on to tonight's event. It's quite an exciting one today because it is celebrating International Day of Medical Physics and also the birthdays of two trailblazing nuclear physicists, Marie Curie and Lise Meitner. So we've got a talk here tonight about research into nuclear medicine. Giving that research is Dr. Christian Diget from the School of Physics, Engineering and Technology and a member of the nuclear physics group here. He's going to talk about the ways that medical physics is already routinely used in hospitals, as well as looking back at the history of medical physics and also into the future of what is currently being done in terms of research today. Christian's the academic lead on the Binding Blocks project, which engages young people with nuclear physics. He's an experimental nuclear physicist, and he tries to understand the nuclear reactions, including exploding stars, as well as new cancer therapies based on radiation and radioactive nuclear isotopes. So I'm going to stop blabbing on for now and pass you over to Christian and let him introduce a little bit more about his talk and what you're going to learn about in tonight's webinar. So take it away, Christian.
Okay, well, thank you so much, Chris, and thanks to all of you for joining us tonight. What Chris was saying, that we're here to celebrate pioneer members of the nuclear medicine community, and particularly Marie Curie and Lise Meitner, who were some of the trailblazers of nuclear medicine in the very early days when we were even just discovering what radiation was all about. My name is Christian Diget, and as Chris was saying, I work in the field that spans from nuclear reactions in stars to nuclear processes that happen inside the body when we attempt to treat or diagnose, for example, against cancer. So I'll move on with my talk. Just to say that before I do that, this work is done at the School of Physics, Engineering and Technology, but also in close collaboration with the Life Science Division at TRIUMPH and other collaborators, including LabLogic and other collaborators that I will mention later on at the end. So with that, let's celebrate medical physics. So what really started nuclear medicine was the discovery of nuclear radiation from materials and moving that into the bodies of people and using it to diagnose, using it to treat cancer, for example. And Marie Curie and Lise Meitner were really some of the key people in discovering this. Marie Curie, for example, discovered both radium and polonium as elements, as well as their isotopes that we are using in treatments. She got the Nobel Prizes in both physics and chemistry, which is absolutely unique. Nobody had done this before, and I believe no one has done this since then. She also pioneered using some of these technologies to develop mobile X-ray units, where actually you could measure, for example, fractures in much the same way that we do nowadays, at least conceptually. The methodology, the radiation came from it in a different way, but it's still the same fundamental principles that underpin it. Lise Meitner, similarly, was a pioneer in medical radiochemistry. For example, for the medical isotope radium-228, I'll come back to isotopes in a moment. She was also essential in the discovery of nuclear fission, which isn't really in nuclear medicine, but actually there are fission products that are used in nuclear medicine as well. So to come back to radium-228, what you see here is a nuclear chart. I will describe in a moment what this is in more detail, but the colors represent different ways in which they decay. And what you can see on the screen is that radium-228 here decays first in 2 decays this way, and in 2 actually many more decays going down as well. Now, the decays going up here are beta decays, where a beta particle is emitted. That's actually an electron. similar to those in the atoms, but much more energetic. And the particles, the decays going down are alpha decays, where a helium nucleus is emitted. And I'll come back to that in much more detail throughout the talk, because the alpha radiation is really critical in relation to treatment of, or future treatments of cancer. So this was the start, but at this time, radiation was seen, yes, as useful, and indeed was used for treatments. It was also seen as magic in some ways, and it was seen as beautiful. So there are weird examples of how it was used at the time, because it was not realized that it doesn't only have the potential to destroy cancers, it also has detrimental potential within the body if it is not controlled properly, right? So of course, a really important part of nuclear medicine nowadays, and in the past many years, has been to make sure that nuclear medicine is delivered in a way that it's safe, right? And indeed, sadly, some of the pioneers, including Marie Curie, died of the radiation exposure because we didn't know, or they didn't know at the time what this was. Got a couple of pictures here exemplifying this. Radiation was used to make fluorescent clocks light up in the dark. That's not terribly dangerous as long as you don't touch it. But it was also used to, for example, put on your skin because you thought it would, because they thought it was particularly healthy or beautiful. It's called radio. In addition to that, and perhaps even probably even worse, there's an example of a radiochemist who put these nuclei into some of the meat that they were eating, or rather that they had left over, to see whether their landlady was reusing the meat on a different day. And indeed, they found it a different day with the radio tracer. I think that's terrible, but and it's not terribly surprising to me that that meat would be reused, but it was, of course, not healthy. The worst thing that one can do with radiation, of course, is to eat it. So that's one of the aspects to consider here, particularly for the type of radiation that we're talking about in this presentation. So let me take a step back before we move on, just in case you don't know as much about elements and isotopes as you really need for the rest of it. This is a periodic table. You may have seen this before. It covers all of the elements from hydrogen right up to oganesson, which was one of the most recently discovered elements. But let me just show you a little bit about what the isotopes of these elements are about. If you take a look at fluorine, this periodic table is fantastic. It comes from the Royal Society of Chemistry, and they give information about each element. So for example, for fluorine, it shows clearly that it has an atomic mass of almost exactly 19, right? That's not a coincidence. That's because fluorine has exactly 19 subatomic particles, protons or neutrons, inside the nucleus of its atom. On the other hand, magnesium has a chemical mass, an atomic mass of 24.3. Now, that's not because there's a third of a neutron or a third of a proton inside its nucleus. It's because there's more than one type of magnesium, and on average, they have a mass of 24.3. So most of it is 24. There's a little bit of 25 and a little bit of 26. 25 and 26 are different because they have one neutron more. So let's take a little bit more of a look at this. So there's an electron cloud around this nucleus. The electron cloud defines what atom it is. The number of electrons is matched to the number of protons, but we can add additional neutrons, right? And depending on which number of neutrons we have added, these are different isotopes of, in the case of magnesium, for example, magnesium 24, 25, 26. And those have different nuclear properties. If they're radioactive, they might have different decays, and we'll come back to that. So we normally show this in a chart, and this is what I showed before. This nuclear chart shows normally neutrons along the horizontal axis and number of protons along the vertical axis. So each row, in fact, in this chart is one element. So you can see hydrogen is at the bottom, then helium, lithium, beryllium, etc. So this is also what was the case for the plot I shared before from the radium-228 work that Meitner did. So radium-228, now it's 228 because it has a total of 228 protons and neutrons. And you can now see that it moves up this way. That means it retains the number of protons plus neutrons by converting one neutron into one proton and emitting an electron. So that's the beta decay. And after that, it alpha decays, bang, bang. And in fact, there are further alpha decays right until it hits stability. So nuclear medicine, now we can introduce that based on the work of Meitner. Nuclear medicine is when we take one of these isotopes and put it inside the body for a medical purpose. Okay, so for example, this could be in order to treat cancer. If we can put it where the cancer is, and I'll come back to that later, if we can put it back where the cancer is, we might get the radiation to kill the cancer inside us. Now, alternatively, if we can measure what's going on, we can use it for diagnostics. And those are the two primary ways in which nuclear medicine is used, either to diagnose illnesses, including cancer, but in many other cases as well, or to treat cancer. So we call these radio tracers in much the same way that the radio tracers were introduced in the meat I talked about before. The radio tracer will go into the body and will move around the body until it finds the right place to attach. And the key here is to have a radio tracer that knows how to find its way. Okay, so let me introduce one such radio tracer that's very simple. The simplest possible, almost, radio tracer is attaching a radioactive fluorine to sugar. So if you attach fluorine to sugar, it still moves around the body as sugar. It is still digested, if you wish, by the cells that use the most energy. There are some parts of the body that use more energy than other parts, but one of the parts that's particularly using energy is cancer cells, because they process food, sugar, they divide. That's what makes them, that's part of what makes them a cancer cell. That's part of why the tumors are problematic. So they use more sugar than most other cells, and therefore they will absorb more sugar, they will absorb more of the fluorine, and therefore more of the fluorine will come into the particular cancer cells. And if you can see where it came from, you can diagnose where the cancer is. It can also be used in many other treatments. It can be used to identify, therefore, metabolic processes inside the body. So how the body reacts, how the body processes different chemicals. So one example of that is, for example, you can use it to scan a brain to understand brain function, and it's indeed an intrinsic methodology in that type of diagnostics of a variety of brain diseases. Okay, so how does it work? Well, it's called positron emission tomography. That's why it's abbreviated PET. Positron emission tomography means that the fluorine, in this case, emits a positron. When the positron is emitted, it's an anti-electron and it moves around the body. It doesn't move very far, maybe a millimeter or two, until it finds an electron and it annihilates with the electron. And from that annihilation, they create two high-energy photons. We call them gamma rays, and they are emitted opposite direction, like this. So if we can detect where they go, if we can detect them out here and out here, we know that they were emitted along this line. If we then detect two here, we know that they were emitted along this line. And if we can do this really precisely for many decays, we can identify where is this sugar or a more active tracer, where is this particularly deposited in the body. So obviously we choose a radiation type, such as the gamma radiation that's very penetrating because we need it to get out of the body in order to detect it. Also, because it gets out of the body, it means that it doesn't harm the body along its way. So the harm is reduced, and therefore we can use it for diagnostics. And we don't have to need to treat in order to justify that we use it as diagnostics. We can use it before we know whether the patient is ill or what illness the patient is suffering from. So if you then have enough of these lines, you can gradually build up this picture. You can see at the top right here, there's an example of just a limited number of lines, about 100 or a few hundred in this plot. If you then do this much more, you can build up a much more detailed picture, such as is the case for this brain scan down here. But can we take this further? And some of my colleagues are indeed doing this. Ruth and Laura are present and are former and present PhD students in this department, in this school. And they work with Professor Dan Watts on developing quantum entangled PET. So quantum entanglement is when two particles are entangled with each other at the quantum level. And the trick is those two photons that come out in PET are already quantum entangled. So they know each other. And even when they hit the detectors, they still know each other. So in fact, what they know is they know kind of loosely, they know how each other spin, right? So the spin of this one is coupled to the spin of this one. So the wonderful thing about this is if you then detect how they turn, that is related to their entanglement. So for example, let's say one of them turns right. What do you think the other one does? Right. So it is a statistical probability distribution, so it does vary, but there is a preference for it to go in a different way. And you might think one or the other. It turns out if this one turns right, it is most likely that the other one turns not left, but either up or down, right? So if you can at the same time measure which way those two turn, you also have information about what has happened in between. Are they still entangled? Are they from the same decay? Can we trust this part of the measurement? Should we reject it or should we use it? So that is what they are currently working on. It is not currently implemented in PET, but this is an amazing discovery that could potentially be implemented in the next generation of positron emission tomography. I think that's fantastic. If you want to know more about it, they have a fantastic description in a video called Improving PET Scanners with Quantum Entanglement. It's already on our Binding Blocks YouTube channel, so you should easily be able to find that. But do take a look at that if you're inspired to do so. But can we treat cancer as well, right? So I said already that we have this methodology of taking a radio tracer, put it into the body, let it move around the body, have it find its place where the disease is happening or where what we need to monitor is happening, and then release its energy and we can use it to diagnose where it is. But there's another part to radiation. There's a more destructive part. What if the radiation deposited all of its energy locally where this radio tracer was placed inside the body, right? I already said that this gamma radiation is not helpful for this because it penetrates the body. It goes out of the body. And the little bit that is deposited within the body is distributed throughout the body, and therefore it is not helpful as a way of treating cancer. What would be helpful, though, is something which has a very short range and deposits most of its energy locally. So, for example, beta decay, many beta decays have a length range of something like millimeters, maybe a few millimeters, in rare cases up to centimeter. That's still a little bit too much for it to be really effective. It means that it doesn't destroy the cells as much as it should along its path, right? This is kind of what is shown here in an image of the or illustration of DNA, where you can see that the beta radiation, it might interact once in a while along the way, but it doesn't interact a lot. The alpha radiation, on the other hand, has high energy and it deposits a lot of the energy in a short period, in a short space, um, within the range of a few cells, 5 to 10 cells. So if we get an alpha emitter as a radio tracer and we get it to attach itself to a cancer cell, and if the neighboring cells are also cancer cells, then that alpha radiation is likely to primarily destroy the cancer cells and not affect the rest of the body. Right. So this is what actually was demonstrated here in this experiment. It was I call it an experiment because it was an experimental treatment. It was not demonstrated prior to this, but they attached an alpha emitter. In this case, AC, it stands for actinium. I will say actinium a number of times from now on. We refer to it as 225 actinium. That's a specific isotope of actinium. It decays via alpha emission, and it is useful in this case, as you will see. So what they did back in 2016, so it's relatively recent, at least for me, it feels recent. In 2016, they tried to attach one of these actinium isotopes to a targeting, we call it a targeting vector, a biomolecule that seeks out the cancer cells, attaches itself to the cancer cell. They knew that this was already a good targeting because they had already used it and tested it with PET. So this was actually one of these sort of more advanced targeting mechanisms, much more targeting than sugar would be. And it was targeting a specific receptor, we call it, on the cancer cell of this type. This was for a prostate cancer. So what they did was, firstly, previously they had tried to attach a beta emitter. You probably can't see the fine print here. Lutetium-177 is a beta emitter. They had tried this already. It hadn't really helped the patient. In fact, the patient had deteriorated in that process. You can see the black dots here are actually from a PET scan of the patient. So you can see that between A and B, after the lutetium treatment, it had actually gotten worse, right? So it certainly hadn't helped the patient and the cancer had spread further. What they then did was that instead of attaching a beta emitter, they attached an alpha emitter to the radio tracer. So it became an alpha emitter targeted treatment using this targeting of the receptor on the prostate cancer cells. And poof, the cancer was gone, right? Now, I don't know what happened much later in this person's life, but already that you can destroy the cancer with a treatment that is not super targeted, right? This was not designed to be targeted at the level that you would expect for a cancer treatment. This was designed to be targeting at the level you would expect or need for diagnosis, right? But they tried it, and based on that trial, prostate cancer is, at least experimentally, now being treated with actinium-225 targeting. Now, the production of actinium-225 is still way too small worldwide in order to actually have this as a viable treatment, broader scale yet. So we need different methodologies for producing these radioisotopes, and we need probably different targeting vectors, as we call them. But the principle is there, right? So the potential is there, and it is fundamentally using the same methods that were pioneered by Curie and Meitner so many years ago, right? The difference is that now we have methods to test how well they target the cancer cells, right? And that is the key, right? That is why it didn't really work 100 years ago, because they couldn't sufficiently target the cancer cells. We can do that now, okay? And that is what is being developed. It's an active development. It is not generally available. it is not going to be available in the next few years, more generally, but it is on the way, right? And I definitely trust that this type of treatment will be under development and will come into treatment more generally at some point in the foreseeable future. So let me show you a little bit more about how this actually works, right? So in the case of actinium-225, it's a radioactive isotope. It's indicated here. It's an alpha emitter. If you recognize it, you can see that actually this is from Royal Society of Chemistry. And you can see in the plot of nuclear isotopes here that actinium decays to francium, decays to astatine, decays to bismuth. That decays, the other decays were alpha decays, and then it decays via beta decay to polonium. Don't know if you remember polonium from before, but I'll come back to that. And then polonium decays to lead, after which very soon it will be a stable isotope. Okay, so there are 4 destructive alpha decays in a row, right? So we need that to get into the cell. So the way we do that is with different types of receptors. You can have a small protein attaching to a receptor. You can use peptide. You can use much larger biomolecules. But the key is, can we then get it internalized into the cell, as you see over here? And let's be honest, that is still an open question in research. Are we internalizing these types of treatments, or are they around the cell, not in the cell? So that is still an open question, and it is still ongoing development. That's part of the reason why it is an active research area where many people now are pioneering that type of treatment around the world. So I said earlier that actinium has 3 other main alpha-emitting nuclei in its decay chain. We call them daughter nuclei because they are, so to say, the daughter in the chain, the next one, the next one, the next one. Might call it granddaughter, and so on. The others were francium, astatine, and polonium. But how do we even measure this? How do we measure where it is delivered? How do we measure the alphas as they are being emitted as we produce the radioisotopes? That's really tricky because I already mentioned that the alpha particles, they have a short range. They only travel about 5 or 10 human cells in length. So it's very short. In fact, it's only about 40 to 100 micrometers, so 0.04 millimeters to 0.1 millimeters. So it's a fraction of a millimeter, about the width of a hair on that sort of scale. So what is the difference? If you want to measure this, what is the difference between these different decays? Well, the difference is in their lifetime. Actinium is the longest lived, and the rest are relatively quick on that sort of time scale. But it's maybe not quick enough that they will be retained sufficiently close to the cancer cell if they decay and they are released from this biomedical molecule that's targeting. So imagine that you've got a biomedical molecule that's attached to something on the cell. So this is on the cell, so it's attached to the cell, and somewhere here is the radio emitter. Okay, so if the radio emitter, this is what I show here, if the radio emitter then emits an alpha particle, what actually happens, or what would typically happen, is that the daughter of actinium would be released from this biomolecule, right? So now we've got a released francium, or next after that astatine, next after that bismuth, next after that polonium. And all of those might be emitted anywhere in the vicinity and not necessarily by that cell. So This is some of the ongoing work that's being pioneered in nuclear medicine, is to try and ensure that the alphas are indeed encapsulated in the cell, or the alpha emitters are encapsulated in the cell so that they stay there long enough. Right. But as I said, they have a very short range. So how do you even measure this? How do you measure the alpha particles? And we're now starting to do that. Not in cells at the moment, but in the production of these radioisotopes. So we're working from York here, we're working with a laboratory in Vancouver called Triumph, and they are producing these isotopes. So we've recently begun to monitor this type of alpha decay directly in the radiochemistry processing. So some of the aspects that Curie and Meitner were not able to do because of the technologies that they had at the time, they couldn't measure this short-range radioactivity directly in the chemical processing. We're now at a stage where we're starting to be able to do that using sensors that are basically embedded into the chemical processing. And we can monitor these decays right from actinium to francium to astatine, right down to polonium, that emits where the final alpha particle is emitted. And indeed, the plot here that I show is super recent data. In fact, it's so recent that if you're watching this online subsequently, you won't see the plot, because I wanted to just show it to you who are here live, because this is data from like 2 weeks ago. But what we're seeing is that we're seeing a range of alpha emissions from actinium, francium, astatine, and polonium within this radiochemical processing, right down to polonium. And if you remember right back to the start of the presentation, indeed, polonium is one of the two elements that were discovered by Curie. So with that, it's been an honor for me to present here tonight, and it's a joy to talk about an area that I find so interesting, but it is an honor to be able to talk on a day where we can celebrate two such significant figures in nuclear medicine. So with that, thank you so much for joining. I hope you've enjoyed it. I hope you have loads of questions, and I know that Chris will do his best to get me to answer as many of them as possible. So thanks to you, and also I should add thanks to our funders as well. Thank you.
Thanks so much, Christian. That was very, very interesting, as I expected it would be. So there are a few questions here, in fact, several questions. And I will warn you that some of them I would say are probably on the biology side, so it'd be interesting to see the perspective on that.
I will do my best.
We'll go back to the original discussion, which was about PET scanners and the way that's emitted.
Yeah.
And we have a question, which is how accurately can the source of the gamma rays be detected? How well do we know the position of these emitters in PET scans?
So that depends on what you mean by this. So the segmentation here are on the scale of millimeter. Okay. But you have this line from which you don't know where it came from along the line. You know something about where it comes from, but not anywhere near to the millimeter scale. So what you need in that case is that you need different angles in order to pinpoint an emission. So I would say down to about millimeter scale is the absolute hard limit. Also because actually the beta plus particle, the anti particle that is annihilating, travels for about millimeter scale, maybe even a few millimeters. So if it already has traveled by a few millimeters, it's kind of already been smeared. So that sets the scale. And in fact, modern PET scanners are pushing so hard on that resolution that it is borderline where it is actually the nuclear physics that is setting the limit for how precisely we can measure that. Yeah.
Cool. Very nice. Got a couple of questions here about the actinium-225.
Yeah.
One is, how do you produce it? You mentioned the fact that there's not much of it, but how
Yeah.
is it produced at the moment? And I guess, is there
Mhm.
any optimism in how we might produce it in the future?
Yeah, so one of the ways is fundamentally the same way that production of radioisotopes happened for in the cases that Curie and Meitner were working on. So, for example, uh, they might be produced in the decay of another isotope, and then when you have that decay for a while, you can chemically separate the product, the daughters, and you can use those. So that was exactly what was done by Curie and Meitner. The difference here is that this isotope will have been produced, that's decaying, will have been produced in a nuclear reaction. So, for example, neutron bombardment, or if I refer to Triumph, that's the one I know the best. They have a very high energy proton accelerator. It's, to my knowledge, physically the biggest cyclotron in the world. It is also a very old cyclotron. It is more than 50 years old now. But they then smash those protons into a target, break the target up at the nuclear level, and through that process create actinium and a whole host of other isotopes. So that is one of the mechanisms. They're working on different reactions, but it's still the same underpinning methodology where you use a nuclear reaction to produce one isotope, and then you use nuclear chemistry in order to isolate the ones that you actually care about. Oh,
Sorry, we've got a couple of questions about how to target that actinium. One specifically, is it a target specific antibody? Do we have
so
someone who's knowledgeable in the
yeah,
crowd today?
so PSMA 617 is an antibody targeting vector. It is also a relatively big molecule, which is why I don't think we know this, but I wonder, therefore, whether that is actually not sufficiently internalized. But some say that it could be. The bottom line, as an experimentalist, is we don't actually have the data on this. So I would say that is still to come. But I would say that with the way that nuclear medicine is moving at the moment, I would say that that kind of information is probably talking within the next 5 years for that kind of information to be available, I suspect. That's guesswork, but I'm very hopeful
Sure.
because there's very substantial investment in this area research-wise.
Yes. It's so important, really life-changing stuff. Yeah. We've got two questions kind of related. Can viruses be used to deliver isotopes? I'm sure I saw a news story about that a few years ago, but I don't know if work's been done on that or if you've got any comment on it.
I think, in short, I don't have any comment on it. I can see that the principle. The trouble with viruses is that they are not that targeting normally. I mean, for example, you may. I certainly recall a couple of years ago when I had different strands of COVID in different years, and they were slightly different, right? So one of them was slightly more chest infecting, some were slightly more throat infecting to me. But that is not the level of targeting that we need. We need something that is really pinpointing the cancer cell. So I would say that it has to be a real biomedical target where the aggressor, if you wish, attaches directly to a receptor that is very specific to the cancer type. Yeah.
Makes sense. There's also a question again. This I haven't heard of. Could isotopes be enclosed in a DNA origami nanobox, or would the decay destroy the boxing, or could it be controlled? That's news to
Well,
me, but
so if you can find a DNA related marker that attaches itself specifically to the type of cancer that you're interested in, it is okay that it destroys the box when it decays, because that is when it releases its destruction, right? So those chemicals that I was talking about before, they are also destroyed when the alpha particle is emitted, right? So the fact that it is destroyed is not in itself an issue. It is about whether you can deliver it to the cancer cell. Yeah.
cool. Is there a risk that the radiation might harm non-cancer cells? How significant is that in the treatment? I
That
guess this is
that
the
is an
advantage.
active risk, right? So, and this is why it has to be sufficiently targeted, right? And I can very
Yeah.
easily use this prostate cancer example as a good example of it. In that case, it was known that the type of targeting they had available, this PSMA 617, was also strongly targeting the thyroid of the patient, right? And there was no way around that. The receptor was present on the cancer cells and in the thyroid, right? So if they wanted to target that particular receptor of the cancer cell, they were bound to also target the thyroid. Now, that wasn't a problem when it was used for diagnostics, because it just means that on the PET scan, they saw, if you look very closely, if you can remember it, um, you would see, or if you review it later, you would see that actually the thyroid was lighting up as well. I think it was included in the picture that I had. So the thyroid was also lighting up in the PET scans. Now, what that means is that that's not necessarily a problem because PET scans are not destructive, right? It just means that the doctor looking at the images need to know that, yes, that part of the scan is not a problem. That's known. It's just the targeting of the thyroid, right? For the treatment, it's a problem. So the treatment killed the patient's thyroid, right? Um, it wasn't considered at the time a risk for that patient. It was considered a an acceptable sacrifice because it was a known risk to some degree, and it was calculated that yes, a loss of a thyroid can be treated with hormone treatments, etc., and is treated for many other patients that have lost their thyroids for other reasons, and therefore that was a calculated, not even risk, I'd say a calculated sacrifice, right?
Yeah.
In other cases, of course, we have to ensure that that risk is minimized, right? And it is done by carefully checking how this targeting vector targets the body. And that is an intrinsic part of any clinical trial of such a cancer drug would be to test where does it attach itself in the body for the patient, right?
Yeah.
Indeed, I can't help mentioning theranostics because really what you need is that you need the same drug for the diagnostics that you have for the treatment. So you need something that is therapeutic, thera, and diagnostic, the gnostic part. So that's why they're referred to, this next generation drugs are referred to as theranostics, because you would then want to use exactly the same targeting when you do the preliminary scan as you do when you treat it and you do when you check whether the treatment has worked, right?
That's so
Um,
interesting. Because
so,
of course, then you
and
know, yeah,
yes, and it's fantastic because the chemistry is the same, right? They are isotopes. They are iso is of the same, topos is the same type. It's the same material, right? So it doesn't it so actinium is not necessarily the best case for it. There are other interesting isotopes of actinium that could in principle be used for this. But there are isotopes under development where you have, or elements under development where you have one isotope of it as being, say, an alpha emitter for treatment, and another element of it being viable as a monitoring device. I mentioned PET. Some of these are not available for PET, but you could, for example, have another imaging type. There's a type called SPECT that's a little bit like PET, but you have less information than PET. So it's not quite as precise as PET, but it's actually super useful as a theranostic complement.
excellent. We have quite a nice open question here, so I will ask it, and then I will also cut you off if you start talking for too long because it's an interesting one. And it's could you explain in more detail how you measure the alpha decay? How do you know where it is? I guess because it stops so quickly, right? Then
Mm,
it doesn't come out, right? So how do
yeah,
you know where it stops? How do you know where the decay happens for alpha
yeah.
emitters?
So the short answer to this is we don't know that yet for the cells. Okay, so that's the short answer. The slightly longer answer is for the chemical processing, we don't need to know in as much detail where it is. So it is super difficult. But what we do know is we have to get it really, really close to our sensor because the range is like this much, right? So in that sense, we know where it is, but it is not actually that is not actually the useful information. Yeah.
Excellent. We've got a really nice comment that we've got someone in the audience who's not a physicist, but found the presentation very enlightening, as did I as a physicist. So that's on the same page. Who actually had a PET scan, and they had to rest in a darkened room before the scan, not even allowed to listen to an iPod before the scan. Yeah, just interesting personal experience of PET scans.
Yeah.
It affects
Yeah.
everybody. It affects us in
Yeah.
everyday life. It's
Yeah.
a interesting one. We've got instead of a full-size antibody, a still specific part of one called an FAB could be used. I think this is a comment about alternative ways of delivering.
Yeah.
Um,
And there are numerous of those under development. I don't know the one you're referring to there, but personally, but there are a number of these different targeting. The trouble with this is that the targeting is sometimes very often specific to a specific cancer type, right?
yep,
So if you have a cancer type where you know the specifics of the cancer, where you know what receptors on the cancer are super specific, and you can design a drug that attaches itself to it, you've got the starting point for a really potential potent alpha treatment.
excellent. Can liposomes been used for delivery? Were they used in mRNA COVID treatments?
Um,
I couldn't.
that so I would absolutely not rule it out, but I don't know.
Yeah, I mean, I think this is it's interesting that
I mean,
as
it
a
just speaks to
yeah,
so that basically it's up to you. It's up to you to imagine what you could do, right? Because there are so many variants in biophysics of how can this, or biochemistry, how can
yeah,
this attach to that, or what is where.
yeah,
And we need many people to use their brain power to identify
yeah.
what are the most effective ways of targeting this.
And
And
it's
I think unless we
creativity and
have these super
imagination.
effective ways, it's not going to go forward. They have to be very specific.
Mhm.
So there will be many failures along the way, right? That's part of it is there will be failures along the way, right?
Yeah.
But there will also be some that are really good at targeting. Yeah.
And I think it also speaks to the diversity and the importance of diversity and the importance of
Okay.
collaboration
Yes.
and
Oh
and
yeah,
and
absolutely.
and physicists
Yeah.
who only stick to what one might consider physics in a very small
Yeah.
box are never going to make these developments and these
Yeah.
understandings. It's people like you and colleagues who kind of cross the aisle, who hold out a
Yeah.
hand of collaboration to people who have different skill sets and different experiences, and
Yeah.
that diversity is yeah, it's so important for the future of science, not just
Yeah.
a sort of a small part of physics.
Yeah.
Could actinium-225 be used in biological circuits for synthetic biology? Again, I wonder if this is for a
Sorry, I
bio.
didn't catch that.
It's Do you believe actinium could be used in biological circuits in synthetic biology? Is that? I mean, it might be a. This feels like we're getting into more biology, but
I I mean, it does release energy, right? It does release energy, right? So from that perspective, I mean, there are radioisotopes have been used as energy generation in relation to circuitry. They are used as batteries. Um, that is part of the powering of space missions and so on. And if that is what it's referred to, then so I mean, in principle, in practice, this type of actinium, it's very short. So medical isotopes are designed, chosen to be short-lived, right? Because if I get a cancer treatment, I want it to be injected. I want it to have time to find the cancer and then decay as quickly as possible, right? So preferably you want something that is longer lived than the biological processing, but relatively short-lived, right? So for example, fluorine-18 is a few hours. Actinium-225 is actually among the longest lived ones for treatment on the market. And that's 10 days. 10 days is very long in a biomedical context. Um, and some of the others are shorter, uh, hours, right? So if it is okay that the lifetime of the circuitry is on the timescale of hours, then possibly. I think in reality it's probably not the right choice. There are other constraints that would be driving the decision in the area you're suggesting. I suspect. I mean, it's not my specialty, but I suspect that it's other um, driving factors there.
Excellent. Yeah, I'll be honest, there are definitely some very knowledgeable people in the audience tonight, and it's great to hear there's some comments and there's some interesting biological
Uh,
statements and things like that. I'm not going to delve too far into those because I feel that it's a place
mhm.
where we don't have an expert to be part of that discussion about in terms of what antibodies might be used in different approaches. So
Yeah,
I'll
yeah, that might make
pause. I'll put a pin in that one, but
yeah,
thank you for your
yeah,
for your comment. That's much appreciated. Um, there's a couple of things more about PET scans, which is
yeah.
interesting. Like PET scans, yeah, they rely on cancer cells using more sugar, but what happens if the cancer's still small and not very active? Would it still be able to pick up and locate the cancer precisely?
Yes, so actually, this has been sugar has been used like this, right? And is still being used in some cases, but it is not used across the board for cancer diagnostics. So I indicated that for the actinium case that I mentioned. Actually, that specific targeting was designed as a PET scan targeting. So these antibodies, for example, you can have antibody targeting in PET scan. So any of the targeting options that you have been suggesting could, in principle, also be PET type targeting. It is only a matter of finding something that is unique. And to the cancer, sugar consumption is somewhat unique, and it is sufficiently unique for some types of cancer, but certainly not all, right? The key is whatever you want to study, whether it's a brain disease or a cancer type or something else, you want something biochemically that is specific to the disease you are studying or the metabolic processes that are dominant in relation to that disease, right?
Yeah. Great. There's two questions I'm going to try and roll into a single question. We've only got a couple of minutes left before I need to round up. But they're both connected to PET scanning. First of all, can you explain the annihilation of the positron with the electron once more? You just broadly go over that again. And
Mhm.
secondly, with quantum entanglement, why is the information useful around the particle response? So how you're describing the fact that the two photons come out and
Mhm.
one scatters up
Yeah.
if the other one scatters left, for example. Why is that useful in
Yeah.
terms of PET scanners?
Okay, so the first is, so if you have a fluorine 18, for example, and it decays, it decays by converting a proton into a neutron, right? So it has something that has positive charge that's changed into something that's neutral. So that positive charge is what is emitted as what we call a positron. That's an anti-electron. This positron, it moves through the body until it stops. When it stops, it finds a neighboring electron and they start to cycle around each other. And once they get really close to each other, just like a hydrogen atom, kind of its atomic level close, once they're close to each other, they combine and all of the energy, all of the mass of those two particles is converted directly into two light particles. So two pieces of highly energetic light, right? Like light here. Um, it is emitted, it's called a gamma ray, but it is a photon just like visible light. It's just much more energetic. Those two are emitted in opposite direction. It's called momentum conservation, where if you have velocity mass going that way and you have something else going this way, they will go opposite ways in order to conserve that. So they go exactly opposite to each other, right? So that is the key part in PET that we know from physics that they go exactly opposite to each other, whether they go this way or that way, they will always go opposite to each other, right? So that's the PET part of it. The quantum entanglement is that as the two photons go away, they also spin. And that spin, it's a little bit complicated. In fact, I wouldn't even call myself an expert in it. So they go opposite ways and they both spin. in different ways. We call it that they're polarized in their spin, and their polarization from one to the other is entangled. So if one is polarized in a certain way, it says something about the polarization of the other. And that is related to how they turn. So let's say this one turns here. What we can do is for the other one, if we mostly accept those that go up or down, we know that we're mostly accepting those that are truly quantum entangled, and we're rejecting most of the ones that are just random coincidences, because there are a lot of decays happening at the same time. So you might have one decay here, one decay here. Both of them have two photons emitted, like this. If we detect those two instead of these two, then if we detect one from one decay and one from another decay, of course, they didn't originate from the line in between the two detectors. They came from two completely different places, right? So it's a way, one of the uses of it is to suppress the types of events that actually are not real, right? And when you're trying to draw thousands of lines on a piece of paper in order to find out where the highlights are, then actually you can get some spurious effects from this type of random coincidences. So that's one of the aspects in which it might be helpful. There are other options for this, but that is, to me, the most obvious, is that you can identify to some degree whether they are real coincidences or random.
It's a noise reduction, right? It kind of it helps.
But it's really important to reduce that noise, because if you can reduce the noise, you can reduce the amount of radiation you need. And if you can reduce the amount of radiation you need, you can get a quicker PET scan, and or you can deliver less dose to the patient, right?
Yeah,
So you can get more capacity in your hospital, and or you can get a treatment or diagnostics with less impact on the
yeah.
patient.
You could have more regular diagnostics. You wouldn't have to wait a certain amount of time
Yeah.
before the
Yeah.
next diagnostic scan. Yeah, no, I think that's really interesting. There are more discussions. I think you've kind of added this that you said spin right and is not just spin up and down. Well, we're talking
It
about
is more
almost a
complicated than
scatter
that.
effect. It's kind of more.
Listen to the talk that
Yeah,
I referred to. They are the experts. They have actually done it. So listen to
absolutely.
that.
There's more questions, but I'm pretty sure we're out of time. In fact, we've overrun by about a minute and a half. So all it takes for me now is to just thank you again, Christian. That was a really interesting talk, generated lots of discussion, and I'm sure people watching, I think you all enjoyed it too, which is great. I'd just like to thank Christian once again for his talk and for joining us and for joining us this evening. Hoping we'll see you at another one of our webinar series, hopefully in the near future. Thanks very much and good night.
Thank you.